Greetings to Dr. Nurashikin. Today, my groupmates and I would like to present to you our research proposal for STF2053 Scientific Communication. The title for our research is the study of antimicrobial effect of Aloe Barbandensis Gel on bacteria that associated with bacterial vaginosis. I will start with the introduction of our research. First and foremost is the background of study. Aloe vera, or scientifically known as Aloe Barbandensis Miller, has been used as a traditional medicine to treat and cure various ailments. Although there are 250 species of aloe vera found distributed around the world, there are only two commercially grown species of aloe vera, which are Aloe Barbandensis Miller and Aloe Arborescens. The significant difference between these two species of aloe vera is the strong latex derived from the bundle sheath cells as well as the clear aloe gel. Our research will be focusing more on the most common species of aloe vera, which is the Aloe Barbandensis Miller. This species is widely known for its anti-inflammatory, antifungal, antioxidant, and moisturizing agent, as well as antibacterial property, which recently have been the main focus due to the COVID-19 pandemic outbreak. Although various research had proved that the antimicrobial activity of Aloe Barbandensis against pathogen, especially bacteria, but there is still lack of research to prove the effectiveness of this plant to heal the infection and soothing the inflammation caused by the overgrowth of certain bacterial species. Several studies had proved that the antimicrobial activity of Aloe Barbandensis against common bacteria found on the skin, such as Escherichia coli and family of Streptococcus. Unfortunately, none of this research have focused on the bacteria that infect the human genital area, especially bacteria that is associated with bacterial vaginosis. Based on the definition explained by Rasubashi et al. In the International Journal of Immunology 2018, bacterial vaginosis is the vaginal inflammation due to the ecological imbalance of the vaginal microbiota caused by the overgrowth of the bad bacteria, in leading to the decrease of good bacteria population, which eventually disrupt the natural balance at the female genital area. The myth regarding the use of Aloe Barbandensis remedy to treat bacterial vaginosis has spread among the community members, in which create a question among researchers whether the claim is scientifically accurate or false. The purpose of this research is to study whether Aloe Barbandensis is effective in the treatment of bacterial vaginosis by understanding the antimicrobial activity of the plant, specifically the leaf part where the aloe gel is embedded. This can be done by exposing the aloe gel of different concentration to both gram-positive and gram-negative bacteria that cause bacterial vaginosis. For this study, we hypothesized that those bacteria that are associated with bacterial vaginosis can be affected by the Aloe Barbandensis gel due to its antimicrobial properties. The higher the concentration of Aloe Barbandensis gel that prevented the bacterial growth, the lower the minimum inhibitory concentration, or MIC reading, of the antibiotic against both the gram-positive and gram-negative bacterial species. Meanwhile, the MIC value of the gram-positive bacteria may be higher compared to the MIC value of the gram-negative bacteria after introducing them to different concentration of Aloe Barbandensis Gel. As for the research objective, we come up with two research objectives, which are to evaluate the antimicrobial activity of Aloe Barbandensis Gel on gram-positive and gram-negative bacteria that are associated with bacterial vaginosis using agar diffusion methods. And we also want to compare the minimum inhibitory concentration or MIC value of the gram-positive and gram-negative bacteria that causes the bacterial vaginosis after introducing them to different concentration of Aloe Barbandensis Gel.
Next, we're gonna discuss about literature review. I'm gonna briefly talk about the title inside the literature review. The first subtopic is essential part of aloe vera. Aloe vera consists of two different parts that produce a completely different compositions of substance as well as the therapeutic properties. Firstly, parenchymal tissue of this plant comprises of thinner portions of the aloe leaf that produce a clear, sticky and tasteless mucus, the jelly-like texture, known as aloe gel. Structural components of aloe vera consist of inner leaf pulp, which are the cell walls, the degenerate organelles and clear, viscous, jelly-like liquid within the cells. According to the researcher, the aloe gel contains approximately 99.5% of water, where the raw pulp of aloe vera have a less water content, which is about 98.5% of water. The further explain is the remaining 0.5 to 1% of solid material contains several compounds.
Next is antimicrobial action of aloe vera on bacteria. According to Encyclopedia of Immunology, it states that antimicrobial action are mediated by the reactive oxygen species, reactive nitrogen species, inhibitor, effector, and protease. There are many methods done to observe the antimicrobial action of aloe vera on bacteria. One of it is this diffusion method. A study proved that this diffusion method is the best method as it is more affordable, fast, and less demanding. Another study done by Fatima Habib and her colleague, they state that Alama blue microtutia are the better method as it is more affordable and better alternative. However, this method requires different standardization and optimization for different bacteria species. Due to this limitation, the Alama blue microtutia are not suitable to be done by the postgraduate student compared to this diffusion method. The fact that this diffusion method are the most favorable method are agreed by the UCAS or European Committee for Antimicrobial Susceptibility Testing. However, these methods are not suitable to observe the antimicrobial action of aloe vera on the anaerobic bacteria species.
The antimicrobial properties of aloe vera have improved in several studies before. In 2013, a study led by Kedanov had proved that the aloe vera extract have a moderate to high level of antimicrobial activity against fungi, gram-positive and gram-negative bacterium. This can be supported by another study conducted a year later after that by different researchers. While in 2017, a study conducted by Darajia and his other friends have found that the aloe vera not only have the antimicrobial properties, but this antimicrobial properties actually occurs due to the presence of the various phytochemicals in the aloe vera extract.
Next, we're gonna discuss about materials and method that will be used in this experiment.
In this experiment, the method that we choose is agar dilution method because this method are suitable to observe the antimicrobial action of aloe vera on anaerobic bacteria. The anaerobic bacteria that we choose is Probacter species, Streptococcus species, Bacterial species, and Gardnerella vaginalis. There are 4 preparations that need to be done before we perform this method. The first preparation is preparation of salt solution. Second is serial dilution of gel aqueous extract. Third is preparation of Wilkins jargon and Arabic agar. And lastly is preparation of inoculum. To prepare the stock solution, the gel extracted from the aloe vera leaf are first minced, homogeneous and dried at 35 degrees Celsius and weighed. Then, 600 mg of the powder are added into 600 ml of distilled water. Next, 1 ml of this solution are added into 9 ml of distilled water to produce stock solution with concentration of 10 mg per milliliter. Next preparation is serial dilution of gel aqueous extract. First, add 1 ml of the stock solution into 9 ml of distilled water to give a concentration of 1 mg per milliliter. Then, prepare 5 test tubes with 10 ml of distilled water in each tube and label as 4, 2, 1, 0.5 and 0.25 micrograms per milliliter. In the first tube, labeled as 4 micrograms per milliliter, mix 19.2 ml of distilled water with 800 microliter of the previous solution. Then, serially transfer 10 ml from the tube to prepare the next solution. The third preparation is preparation of Wilkins jargon and Arabic agar. To prepare this agar, 43 grams of Wilkins jargon and Arabic agar will be suspended into 1000 ml of distilled water and heat to completely dissolve the medium. Then, to dispense and sterilization, autoclave this agar at 15 psi for 50 minutes at 131 degrees Celsius. Then, let the agar chill to 50 degrees Celsius and add 50 ml of defibrinated blood into the agar.
Last but not least is the expected outcome. For this experiment, we expected that the Aloe Barbandensis gel will have a significant effect on the bacteria that causes bacterial vaginosis, which are the gram-positive bacteria, Gardnerella vaginalis, and Streptococcus species, as well as the gram-negative bacteria, Preventella species, and Bacterial Fragilis. We also expect that the higher concentration of Aloe Barbandensis gel will prevent most of the bacterial growth and thus lowering the MIC value of the antibiotic, in this case the Aloe Barbandensis gel, for both bacteria species. Lastly, we also expect that the gram-positive bacteria will have a higher MIC value compared to the gram-negative bacteria when exposing them to different concentration of Aloe Barbandensis gel. So, why this study is very important? The significance of this study is to evaluate the antimicrobial activity of the Aloe Barbandensis gel in order to understand how this gel can effectively act as an antimicrobial agent that may have the potential to replace the other drugs or chemical compound medicine for the treatment of bacterial vaginosis. Furthermore, by manipulating the concentration of the Aloe Barbandensis gel, it is possible to determine what concentration of the extract that can affect the bacteria associated with bacterial vaginosis. Furthermore, this research may also help to find other alternatives by making use of the traditional remedy in order to treat the infection like bacterial vaginosis. Therefore, it is highly essential to learn more about the plants that have the medicinal property like Aloe Barbandensis Miller in order to confirm that the gel extracted from the leaf could be utilized for the treatment of an infection associated with the genital area. Thank you.